Improved Parkinson's disease testing. Just some background. Parkinson's disease is a generally late onset disease that leads to loss of control of hands and other extremities, including symptoms of shaking and stiffness. The disease is almost purely genetic, with very few environmental factors contributing to Parkinson's in observed patients. Only 15% of Parkinson's patients have a family history of the disease, so heritability is still questionable as if it has a direct correlation. The PARK7 gene is often associated with Parkinson's, producing the DJ1 protein that aids in the shaping of newly produced proteins according to SNPpedia. Parkinson's is a complex disease in that many of the observed genes of high frequency in Parkinson's patients are not understood and what a lack of or excess of proteins do to cause the tremors along with the lack of dopamine. So 23andMe, they test for the G2019S variant and the LRRK2 gene and the N370S variant in the GBA gene. Parkinson's affects the production of derridin, important to maintaining the strength and structure of the cytoskeleton of cells. In a Parkinson's patient, the LRRK2 gene produces little to no derridin, but scientists are unsure as to how it is linked to the symptoms of Parkinson's, but observe a low rate of derridin in Parkinson's patients, according to Medline Plus. By having the G2019S variant of the LRRK2 gene, there's a 30% chance that the person will develop Parkinson's, and the chance only increases with age. There are other genes associated with Parkinson's not tested by 23andMe, including the SNCA and TMEM230 genes. Designing our test. There are approximately 16 different SNPs that affect Parkinson's disease. However, 23andMe only tests for 2 variant SNPs that are confirmed to have an impact on Parkinson's disease. These are on the LARRK2 and GBA genes. One variation is the RS and is not tested by 23andMe. This variant specifically affects the EIF4G1 gene. This affects the cases of familiar Parkinson's disease and is very rare and is a dominant allele. It is not a protein, but it does affect the chances of late onset Parkinson's disease. Alleles AA and AG have shown very strong correlation to leading to late onset Parkinson's disease. It is expressed in brain cells where Parkinson's disease will mainly act. If you have this mutation with family history of Parkinson's, it is almost a certainty that you will develop late onset Parkinson's, according to omim.org. First, we will start with a PCR in order to replicate the DNA to be examined on a SNP chip, because we will only be examining SNPs that lead to Parkinson's. The test will consist of a SNP chip that tests for both the variants that 23andMe tests for on the LARRK2 gene and GBA genes, as well as the untested variation of RS.
Our test has one main benefit over the test from 23andMe, and that is the examination of variant RS1121764450. This variant, when a patient tests positive for it, means that there's a very high chance that the patient will develop late onset Parkinson's disease. It's a familiar trait, but this is our main benefit, is that we will test for this and 23andMe does not. Another reason is due to cost. It will cost roughly about the same to take both tests because we're using a SNP chip, and we can simply add some more tests for that variant onto our SNP chip. There are 3 potential limitations of our test, all stemming from a similar issue. Number 1 is that inherited alleles are not super accurate in predicting Parkinson's disease. About 85% of Parkinson's disease cases are sporadic and not directly linked to inheritance. The second is that there are about 18 to 20 genes currently known to impact the chances of Parkinson's disease, meaning that there may be more that this test does not tell you about. And because it is a SNP chip only looks for what you code it to look for. And so in this case, we would not be able to see any other genes. And then the third one is that the basic risk without the variant is relatively low, about 13 in every 100,000 people in America. And this test for our variant would only be necessary for those with family history, seeing that it's a familiar trait that it becomes a dominant gene, guaranteeing the late onset of Parkinson's disease. So this is a limitation because it does not work with in cases where you have no family history, so sporadic cases. And with any genetic testing, there's always ethical considerations. And the main ethical consideration for our test is going to be that if there's a positive test, it would be a warning sign about Parkinson's disease, and it would likely cause a lot of stress and an emotional toll. There's no huge financial burden that is associated with treatment because there currently is no cure for Parkinson's disease. However, there may be a cost associated with preparing facilities for once the symptoms onset to make caregiving easier and quality of life better. And the emotional stress would likely be much higher for if you test positive for our variant, because that means that with near certainty you would be developing Parkinson's late onset. And then as the testers, the ethical preparations we need to take are making sure that every patient is aware and acknowledges these potential stresses in their life before receiving their results from our test. Thank you.